Good afternoon. I'm trying not to be late, but I think I have too many slides. So, my name, I'm from Lyon, in France. I'm representing an innovative technological platform. So it's a governmental founded platform, nothing private. And we are founded by the government to help the companies and the laboratory from France and Europe to make innovation with additive manufacturing. So I will go fast in the first ones because I want to go into the detail of what we are doing, but this is very administrative part. So we have a large budget for each year, partially funded by the government and the other part half by the private contracts and the collaborative projects. Just the network of the companies that are working with us currently. So as you see, it's quite a lot. I can see the screen. So just to show what are the offers we have. So we are working in the in the bioprinting for sure. I will talk more about that later. 4D, high resolution, have few slides also. Holographic, I will not talk about that. It's a technological development. And ceramic, we also have a large part of ceramic fabrication facilities, but I will not talk about that today also. So, bioprinting. So we have developed for the last five years different bioprinting for different applications. So this one was for skin printing with the Lab Skin Creation. It's a company from Lyon, so they asked us to develop something with them. So we developed the process, but also the machine with a French company and so on. I will talk a lot about the skin later on. Then we have R&D. Pure R&D machine with a lot of control inside, just showing you one particular printing here. I want to emphasize something about these printings. What we try to do real 3D printing with cells, not just cell ladder or little square. So this, for example, it's implantable collagen and sugar material in which you can disperse cells, living cells. And we are able to do this kind of print. I mean, high resolution, nicely defined prints, wall cells with implantable materials. And for us, that's the key. I mean, being able to print something which is not implantable, then you're stuck somewhere between your application and the clinical application. So that's the issue here. Then we have this robotic arm, which I will talk about later also, which we think will bring the bioprinting inside the surgical theater, but I will talk about later. Ceramic printing, I will go fast, we don't talk about that. Inkjet, fuse deposition, that's the basic we use to modify the other machines and develop the tools. DLP, we use that a lot for the 4D printing, meaning some printing with biomolecules inside, active molecules. I have just one slide, and the high resolution, this one, which is just starting to have application in the area of bio. Just to show you what have to be in a lab to make some innovation in the 3D for life science. So we have rheologists here, pure rheologists, polymers. We have specialists of the biomolecules. We have specialists of high resolution. So it's more like photopolymer specialist. Here also photoinitiator polymer specialist. And then we have specialists of bioreactors, tissue engineering, and so on. And all these people, they have to be together in one place, otherwise it's too slow to work. So one first application that we developed for the last 4 years, it's a human skin, and we are trying to do this complete laminar organized human skin. So we did it with a company in Lyon, and that's the process. If you look on the left-hand side, the idea is that you want to grow these different layers one after the other, but this layer they're maybe not compatible in terms of maturation of the tissue and so on. So you need to be able to do this process, printing, consolidating, that's chemical or biochemical processes, whatever, maturation, then you have one first layer, mature, like a dermis. And then you need to be able to print on top of this, to print the epidermis, for example. And then print also at the bottom to print the hypodermis. And all these different layers, they have different maturation protocol, different culture condition, and so on. So that's not an easy task. So the beginning was easy, let's say. It was not that difficult. We print the hypodermis, and then we print the dermis, 2 types of cells, not so difficult. That was 2016. And we had this nice picture, so I have lots. It's a picture contest. I have so many that represent the skin. So all these differentiation factor, markers and growth markers, they are here about human normal skin and 3D. So we demonstrate that it was easy and not easy but feasible. So we have a lot of different markers. This was published years ago, I think. And then more pictures about distribution of the cell, living cells, the different markers, and so on. And then we have to move to a more complex system. For example, the epidermis is not only keratinocytes. You need to be able to put some other cells inside. So that's where we move to the melanocytes also inside the second ink. So we managed to do that. So now we have dermis, epidermis with melanocyte inside. So you can tan this under the UV. They change the color and so on. And then you need to go to the hypodermis, because if you think about regenerative medicine, if you think about macrovascularization and so on, this took place in the hypodermis, and that's where we want to go. We are not so much interested in the drug testing or cosmetology testing and so on, but more in the grafting of this substitutes on top of severe burns people. So we want to have this part. So we managed to do that based on ADC. So there's a little bit of cellular biology before, fractionation and differentiation and so on. But we managed to get this nice hypodermis. So if you look here, so you have hypodermis and dermis, and then on top of this, the epidermis far from here. So now we have four different cells in this model, and we approach slowly but surely to the complete skin. So all this result about hypodermis and melanocytes, it's not published, just for you today. So that's where we are. So we are here. 2015 we were here, just dermis, and we tried to get this macrovascularization in the next year. So that's very important for us. It's very important according to the reimplantation of this for the clinical application. And I put 2 things in red here. So we have this special project. I'm going to talk about the in vivo bioprinting that started this year, and also we have this very important topic. I have this in all of the different tissue, which is the bioreactors for this. Specifically for this bioprinted tissue, and this is very important for us. So about the 3D in vivo printing. So that's a different concept. That's for skin also, because we want to be able to have access to, so I think I have this little mouse in my pocket. So what you see is something printing on this mouse, sorry. So the idea here is that we have this robotic arm, and we graft on top of this guy this bioprinting head. And the idea is to get this medical imaging, then the biopsy inside the surgery theater. We get the biopsy, we make the ink, and we print back the ink on top of the burn. So that's proof of concept here. We are working on mainly on this part here, because we want to do that in the surgery theater, not going out, keeping everything inside. And we are working a lot on making this ink medical grade ink, because for now the compound were not medical grade. Anyway, we'll move fast. And then we have a second project, which is human cartilage. So we work for that for the last 2 years with a lab in France. So the idea here is not primary cells, but we are working with stem cells. So we had a proof of concept of these cartilages in 2016, and then this year we move very fast to pure, very mature cartilages with nice collagen synthesis inside the collagens, also nice proteoglycan synthesis. And the thing is that, and we were quite surprised that the complete, so it's 1 centimeter thick. So the complete print is full of this neosynthesized matrix, and we are very keen of this. So this is also non-published results. So we're going to try this in vivo implantation next months, and we hope it's going to be nice. So for next, so one more time, 15 we had the proof of concept, and now we are around between these two, and we don't know yet what are the mechanical characteristic of this cartilage, and we need to go through that to be sure that it's something interesting for regenerative medicine. But we still have the in vivo validation in the next year. And then we have another collaboration with a French institute in Paris, trying to print some large tissue with tumor inside. Because one more time, what's interesting in the 3D bioprinting is that you are able to make some large things with complex shapes, not just teeny square of something. So the idea here is that we are using some prostate cancer cells, and this guy in Paris, they know how to make some spheroids with them, and we are printing these spheroids in large objects. And the idea is to see these spheroids grow inside our objects and make large tumor, and make something that is more like what you have in vivo than something that you have in a small spheroid like this. So first results looks like it's working. So we have growth of this spheroid tumor. So we have merging also of these spheroids, and that's nice for us because meaning that the building block of one spheroid can be used to print something. So it's like a building block, and then you assemble them inside an object. And the beauty of this is that you can also co-print with the fibroblast, so with human tissue, but healthy human tissue, and then you can have something that looks like a carcinoma inside. So here it's spheroid spread inside the fibroblast tissue that you have printed. So you can co-print them and try to mimic something. And then the next step will be to have tissues with vascularization inside, and then being able to really recapitulate something that is just like what's happening in the tumor. I mean, the tumor is a lot of blood vessel inside. So the idea is to have something with high vascularization inside. So then, going to the vascularization, because I mentioned that we want to make large objects with tissue inside and so on, but you maybe know that if it's too large, then nothing can go inside and the cell died. So we have a strong part trying to figure out how to make this blood vessel, these large blood vessels, and how to make them connected to the outside world, because making a tube it's easy, making a tube with connection it's not easy. So this is one. I have few slides about that. One strategy we have with diffusion of catalytic reaction inside shapes that leave the inside free for flowing. We have sacrificial also systems that can be filled afterward with biopolymer and seed with cells. And we have this interesting part, which is trying to produce at the same time a plastic part and a biomimetic, biocompatible part with hydrogel. So here we are using the DLP. DLP, you can make some photopolymerization of acrylite. This is your plastic part. And then because you are using the same chemistry between the hydrogel and the plastic part, then you have sticking here very hard, and you can flow You can plug something in your plastic, flow something in your hydrogel, in which you have cells on the top, on the wall, and then you are able to have one vessel connected to the outside world through the plastic, and that's a real challenge. I mean, very, very difficult to have biological and plastic, printing the biological before the maturation of the tissue, and being able to plug something, because when you print the hydrogel, they are very, very fragile, and it's very difficult to plug them. And then we have this part about the bioreactor. So it's an internal project, so we try to figure out, is it feasible, is it interesting to print the bioreactor according to the shape we have in terms of bioprinting. So this is the bioprint. This is the bioreactor, which is print, and we try to flow inside to see what is the impact of this flow, of the shape of this reactor, the shape of this structure here on the organization of the cell, and of course trying to make some blood vessel inside. So just preliminary result, not published yet also. So this is what's happening after 10 or 12 days of flowing inside. So we had a structure at the beginning with holes and so on, and we start to have this UVEC very nicely covering this tubing and making some vessels. So for now, nothing sure about how the vessel is working and so on, but that's the concept. I have 3 more slides, I think. Silicon printing, because we are using all the different techniques, all the different materials that can be implanted or grafted to people. So this is silicon printing. This is a project with a company, Prototype, which is doing some very complicated implant for trachea. So if you look at this implant, I mean, the inside, it's that side. The inside is very complicated to mold and to print, basically. And that's where we are working. We are working on the formulation and the processes for this printing of silicons, in order to be able to make these shapes, very complex shapes, to do in silicon, with implantable silicon, very difficult for them. And finally, we talked about that this morning, so I will go fast. We have a special activity with the hospital for pre-surgery, mainly with small kid surgery for models. So that was our first one with a tumor here in the Broca. This is one of the last. And here, what we try to do, we try to integrate what we know about the silicon printing, making the lung, for example, more realistic, and what we know about the plastic and the segmentation, but you don't really care about that. The idea is that we try to integrate all the development we have in the other projects to help these parts in terms of new component for these models. High resolution. I don't know how many times do you have? Out of time. Nice. High resolution. I just want to give a word about that. So we are working with a company in France, Microlite. You have almost the same here, Nanoscribe. High resolution 2-photon polymerization. We are using this to increase the fidelity of our skin model. So the idea here, we are working with this company which is making some segmentation, but from confocal microscopy. We are trying to recapitulate the dermoepidermal junction, which is a very complex structure. And here you have a printing of this junction in collagen, and the idea is to seed some cells on top of this collagen structure, which is the junction you have between the dermis and the epidermis, and try to see if we can make new models of skin, pure model for skin, and try to recapitulate this junction. And then, just to finish, because it's more than bioprinting today, we also have a new platform in Lyon, which is called Axel One Printing, which is part of the 3D printing, the 3d.FAB. And we are focusing on technical plastics and technical polymer for the 3D. So this platform is doing development in terms of formulation and processes in order to be able to print some highly technical charge polymers for space application and so on. And we are doing this with these people. So it's just launched. And I'd like to thank you for your attention and happy to answer your questions.